Hey team, welcome to the screencast on aerobic respiration. So we have covered already the whole anaerobic process. So we have glycolysis there. Remember, we had made a net of 2 ATP in this process, and then during fermentation we regenerated our NADH to NAD+. Today we're going to be talking about the aerobic pathway. And remember, this is in the presence of oxygen. And I'm not really going to talk very much about glycolysis at all, because the glycolysis that happens in aerobic respiration is basically, well, it is exactly the same process as the glycolysis in anaerobic. We have 2 ATP made and in the exact same way. And today we are going to cover the preparatory steps, which I'm just going to call the prep steps from here on out because it's a little easier to say, as well as the Krebs cycle. That is what the screencast will be about today. So moving forward, just to give you a little reminder about glycolysis, here I'm going to pop. So here, this is a screenshot of our anaerobic respiration, and I can just wipe this out and say aerobic respiration because glycolysis is exactly the same. Now, as you'll remember, in glycolysis, we split one glucose molecule and we ended up with 2 pyruvate molecules, and in the process we created 4 ATP, but remember we spent 2 at the beginning in our investment phase, so on net we made 2 ATP. And then we also made 2 NADH, which in the process of fermentation we regenerated to NAD+. But in aerobic respiration, these 2 NADH are actually very useful. So we are not going to regenerate them in fermentation. We're actually going to keep them for later. These NADH molecules are holding high energy electrons that are going to be used to do useful cellular work later on in aerobic respiration. Okay, but moving forward, we're just, again, talking about the prep steps today and the Krebs cycle. So let's jump on into the prep steps. Okay, so you remember, after glycolysis, we end up with 2 pyruvate. And glycolysis is happening in the cytoplasm of the cell, and these 2 pyruvate are going to move into the mitochondrion for the prep steps. So I'm going to write mitochondrion right here, and it's moving in there for the prep steps. They're called the prep steps because they're preparing these this compound, these 2 pyruvates, for the Krebs cycle. So we've got our 2 pyruvates moving in, and I'm just going to copy these guys, paste them down here. So remember, we've got 2 of them. And then there's another molecule here called coenzyme A. It's a coenzyme, meaning, so this coenzyme you can think of as a helper molecule. It's called coenzyme A. So I'm just going to abbreviate it here and call it CoA. And then in addition, we also have 2 NAD+. And these things are all going to combine in a reaction to form a molecule called acetyl-CoA, and this is a 2-carbon molecule. So I'm just going to grab, so we have 2 acetyl-CoA, and then we also have 2 CO2. So you wonder where the other carbons went. Well, it's broken off of the pyruvate molecule and released as CO2. And then in the process, we also give NAD+, or 2 NAD+, high energy electrons, and they become NADH. So now this NADH molecule is holding 2 electrons. So there's a lot of energy in these pyruvate molecules here. And by breaking them down, by releasing the CO2 and giving NADH, this high energy electron, we're going to use it later to do work. So I'm just going to label these things right here. We have acetyl acetyl-CoA, and this is CO2. So then we're going to save, so this is the whole thing, these are the prep steps right here. We're going to save this NADH, or these two NADH for later, and this acetyl-CoA, which still has a lot of energy in it, is going to go on to this process called the Krebs cycle. Oh, and just one more note here. Know that this pyruvate has been oxidized into acetyl-CoA, and this NADH has been reduced. So it's gained a high energy electron, and pyruvate has lost the high energy electron in the process of making acetyl-CoA. So we're slowly here reducing, or we're slowly oxidizing our glucose molecule. We're slowly stripping away energy and storing it in other places. And instead of just burning our glucose in the bonfire, or we are slowly, it's like feeding the fire slowly instead of just igniting all the gasoline in the engine so it explodes in a big fiery burst. We're slowly stripping it away to do useful cellular work. Okay, now let's talk about the Krebs cycle. So as you know, for every one glucose molecule, we're getting 2 acetyl-CoA coming out of the prep steps. But I'm just going to talk here about 1 acetyl-CoA going into the Krebs cycle. But you know here that actually 2 acetyl-CoAs are produced for every molecule of glucose. But it's just a little easier to talk about 1 molecule going through the cycle. So the Krebs cycle is a cycle, much like the C3 cycle, except here instead of building a molecule, we're going to be breaking a molecule down. And we're going to be releasing CO2 instead of incorporating it into our organic molecule. You might also hear the Krebs cycle called the citric acid cycle. So much like the C3 cycle has many different names, the Krebs cycle has a few different names as well. So don't be confused if you hear it called the citric acid cycle. It's just the same process. Okay, so we have an acetyl-CoA entering our cycle here. And it is going to bind to a 4-carbon molecule that you've actually seen before. It's called oxaloacetate. So acetyl-CoA comes in, binds to this oxaloacetate, and then makes a 6-carbon molecule called citrate, which can dissociate into citric, and it's an acid, so it can dissociate into citric acid, which is where the citric acid cycle name comes from. So I'm going to draw those molecules coming together. So we've got this 4-carbon molecule binding to our 2-carbon molecule to form this 6-carbon citrate molecule. And this is a really high energy molecule, this 6-carbon citrate molecule. And so we can use it, we can strip it down and extract some of that energy from it. And we do that by taking our NAD+, which is going to come into the cycle, feed into the cycle, and in doing so, it's going to bust up our citrate molecule into a 5-carbon molecule. So in the process of busting up this molecule, we create a 5-carbon molecule, and we also release a molecule of CO2. So this CO2 molecule is released, and also we create an NADH molecule. NADH here. This is holding here a high energy electron. So we have this 5-carbon molecule, and then once again we have NADH coming into the cycle, or sorry, NAD plus down here, feeding into the cycle, and again busting up this 5-carbon molecule into a 4-carbon molecule. So then essentially the same thing happens again. We're going to add this NAD+, and then we're going to bust off a CO2 molecule, and we're going to create NADH in the process. So we're slowly stripping away the energy from the citrate molecule and transferring that energy into NADH. And I'm just going to label this here. This is, you should know this carbon is a CO2 actually. And this carbon is also a CO2. So you know when you all breathe out you exhale about 20 respirations every minute if you're in relatively good shape and at rest. Well, this CO2 is exactly the CO2 that you're exhaling. That's where your breath, your exhale, comes from. The creation of this CO2 in the Krebs cycle. Okay, so then you're left down here with this 4-carbon molecule, which you don't need to know the name of, but you should know that it's a really high energy molecule. And so this oxaloacetate that we are going to regenerate in the end is a really, really low energy molecule. And so this molecule here, this 4-carbon molecule, still has a lot of energy in it. And we can actually perform another substrate level phosphorylation of ADP and inorganic phosphate. Here we add this into the cycle, and out pops an ATP. So you know the whole point of making of this cycle is to make ATP, and we're going to use the energy stored in these NADH molecules to do that later. But we can do some substrate level phosphorylation here. So again, this 4-carbon molecule loses a little more energy, but still, it's got more. So as it goes around the cycle, we have another molecule called FAD. This molecule FAD+, which is just like NAD+, it's a high energy electron carrier. It feeds into the cycle here, and it pops out as FADH2. FADH2. So again, it's holding high energy electrons for us. And then right before we form oxaloacetate, we still have a 4-carbon molecule with some energy. So again, we feed another NAD plus into the cycle, and we pop out an NADH. Oops. And then, at this point, we regenerate oxaloacetate. And you should all know that this process is, there are a lot of enzymes that come in, and they're catalyzing this reaction, this cycle. But this whole thing is a cycle, the Krebs cycle, much like the C3 cycle is a cycle. Okay, so let's look at the cycle for a second and do some accounting. As you see here, we had one acetyl-CoA feed into the cycle, but you know that we have 2 for every molecule of glucose. So I'm going to go up here for a second. So if we talk about 1 molecule of glucose and what we're getting out of that 1 molecule of glucose in glycolysis, we produced 2 ATP for each molecule of glucose, as well as 2 NADH. In the prep steps, we produced 2 NADH. And then, in the Krebs cycle, for one turn of the cycle, we produce 3 NADH, as you can see here, 1 FADH, and 1 ATP. So that's 1 turn. Whoop. But as you know, for every 1 molecule of glucose, we're going to get 2 pyruvate and 2 acetyl-CoA. So you can imagine this cycle turns twice for every molecule of glucose. So really, in the Krebs cycle, we create 6 NADH, 2 FADH2, and 2 ATP, as well as 4 molecules of CO2. But you can kind of think of that's a waste product, not really something that we're counting as towards our 1 molecule of glucose. Okay, so this is what we're getting out of 1 molecule of glucose. This is our counting process. Okay, in your next screencast, I'm going to talk about where all these high-energy electron carriers, these NADH and FADH2 molecules, go to release their energy. And the last thing that I want to tell you all is that you do not need to memorize this whole cycle. What goes in, what goes out, the exact numbers. If we ever have when we have a test or an assessment on this, I will always give you a picture of this. What I really want you to focus on here is understanding the process and being able to interpret what happens. So again, don't memorize any of this. I just want you to understand, if you're given this diagram, you should be able to interpret it. Okay, that's it. Thanks for tuning in on aerobic respiration, and I will see you all in class on Tuesday.